Welcome to the Bodhi Academy. My name is Shrutika and today we are going to continue with our lesson from standard level biology, that is biomolecule. So in this video, in our previous videos, that is part 1 and part 2, we have seen the different kinds of biomolecules. We have seen how the biomolecules are classified into different types, and we have also seen in detail about each of the biomolecule, that is carbohydrate, nucleic acid, proteins. Then we have also covered the different structures of proteins, and also we have covered the last one, which was the nucleic acid. So today what we are going to study is enzymes. So in this part we are going to deal with enzymes, and we will study how enzymes work in our system. So what are enzymes? So enzymes are commonly proteinaceous substances which are capable of catalyzing chemical reaction of biological origin without themselves undergoing any change. So they are commonly called as biocatalysts. So enzymes are basically proteins. Enzymes are what? Proteins. So they are basically proteins which will help in increasing the rate of the reaction. So we all know that our body undergoes various kinds of reactions, and those reaction needs to be fast. So basically what are enzymes? They are proteins which will catalyze as in it is increasing the rate of the reaction without itself going under any changes. So enzyme is not, and the structure of the enzyme is not getting changed. So without it itself not getting changed, it will increase the rate of the reaction, that is, it will catalyze the reaction to form the product. So this is about enzyme and what is the meaning of enzyme. So now let us see what is the nature of enzyme action, as in how the enzyme acts in a reaction, as in how enzyme will be like involved in a reaction. So each enzyme has a substrate binding site in its molecule so that a high reactive enzyme substrate complex is produced. This complex is short-lived and dissociates into products, and the unchanged enzyme with an intermediate formation of enzyme product complex. Okay, so what happens? So enzyme, so if this is a enzyme, okay, if this is an enzyme, and it will have, see over here, enzyme has a substrate binding site. So this is an enzyme, this is the substrate binding site. If a substrate, so this is like a lock and this is like a key. So this substrate will come, this is a substrate, okay, this will come and bind to the enzyme and will form what? Enzyme substrate complex. Okay, then from this complex what will be formed is the product. Okay, what will be formed is the product, and then after the product is formed, because of the enzyme substrate complex, the product will leave the enzyme the site, the binding site, the product will leave, and you will get your enzyme separately and you will get the product from the enzyme substrate complex. So this is how enzyme acts on any substrate, and this is how the nature of enzyme action is, as in how the enzyme will work on any reaction. So first it will form enzyme substrate complex, then it will form the product, and then enzyme and product will be separated. So this is the nature of the enzyme action, or how enzyme action takes place. So what are the steps in this reaction, as in how this reactions are taking place. So catalytic cycle of an enzyme action can be described in the following steps. So what is the first step? So the first step is the substrate binds to the active site of enzyme, fitting into the active site. Okay, that I have shown you all now over here, that so this is an enzyme. the substrate will bind to the active site of the enzyme. The binding of the substrate induces the enzyme to alter its shape. So whatever the shape of the substrate is, if it is this shape, then the enzyme will change its shape to this, so that this fits into the active site of the enzyme, or to the site of binding. Okay, so if this is substrate, this is enzyme. If the substrate is of this shape, then the enzyme will change its shape to this, so that this fits into the enzyme substrate complex. So then what is the substrate? The third step, the active site of the enzyme now is closely proximate to the substrate, breaking the chemical bonds of the substrate, and new enzyme product complex is formed. So then after the reaction takes place between the substrate and the enzyme, what is formed is the product enzyme complex. So this is the substrate plus enzyme complex, and this is what? Product plus enzyme complex. Then what happens? The enzyme releases the product of the reaction, and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle again. So then what happens is, your enzyme will leave the product. And we all know that a chemical reaction, whenever a chemical reaction is taking place, what is formed? A new product is formed. And this is how the enzyme is helping in formation of that new product. So then after the enzyme substrate complex, the product will be formed with the reaction of enzyme and the substrate. Product will be formed, and once the product is formed, the enzyme will release that product, and then that enzyme is now ready for another substrate to bind to it and continue the catalytic cycle. So this is how the enzyme activity starts, or how the enzyme acts upon any reaction in which it occurs. So now let us see what are the factors which influence the enzyme activity. So enzymes are very sensitive to temperature, pH, etc. because they are what? They are proteins. So basically proteins are very sensitive to temperature, and any change in the structure of the enzyme will change its activity. So let us learn what are the factors which are influencing the enzyme activity, because if the shape of the enzyme changes, then the enzyme will not be there in the state of action. Okay, so now let us see what are the factors which will influence the activity of the enzyme. So the factors are temperature, pH, and concentration of the substrate. So over here, first factor is the temperature. An enzyme is active within a narrow range of temperature. Temperature at which enzyme is most active is called as the optimum temperature, and the enzyme activity decreases above and below this temperature. Okay, so there has to be a optimum temperature. For example, it can be 60 degrees Celsius. So this will be like the optimum temperature for the enzyme activity. Below this and above this temperature, so this if it is 60, this will be the temperature at which the enzyme will show maximum activity, and below this temperature and above this temperature, the enzyme will not show its activity, that it will not help in the formation of the product. So this is how temperature affects the enzyme activity. So there has to be an optimum temperature at which an enzyme works. So over here, so this will be, if we look at the figure 9.5, figure B, this peak side will be the optimum temperature. Okay, so this will be the optimum temperature at which the enzyme activity will be maximum, and it is gradually increasing. See, so below the optimum temperature and above the optimum temperature, the enzyme activity will decrease. See, over here it is less, and over here it is less. Over here, this is the peak site where the enzyme will be very active. So this is how we can see, represent the activity of the enzyme. Second is the pH. So every enzyme has an optimum pH at which it is maximum active. Most of the intercellular enzyme work at a neutral pH. Neutral pH as in if the pH of the solution is 7. So 7 is the neutral pH. So 7 is the neutral pH. Okay, and if this is the, see over here, the pH is showing the peak. So I can say that this is the optimum pH. So if it is 7, then the enzyme activity will be more. If it is less than 7 or more than 7, the enzyme will show less activity. So this is how we can say that pH, optimum pH is very important for the enzyme activity. Then comes the concentration of the substrate, as in concentration of the substrate, as in how much substrate is present in the solution for the enzyme to react upon. So increase in substrate concentration increases the rate of the reaction due to occupation of more active site by the substrate. Okay, so if substrate is more, it will occupy more active sites and the reaction will be increased. So see over here, as we increase the, in this graph we can see that as we increase the amount of substrate, the activity of enzyme is more. Why? Because there will be more binding sites for the substrate. So more the binding site for the substrate, the reaction will also be increased. So this is how the factors, that is temperature, pH and concentration of the substrate, influences the enzyme activity. So this was about enzymes. So in our today's video, what we have learned is enzymes, then the nature of the enzyme action, how the enzyme action takes place, the various steps, and we have also seen what are the factors which affects the activity of the enzyme, that is temperature, pH and concentration of a substrate. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe.